you very much for coming. I want to thank the administration of the Radcliffe for this wonderful year. Thank you to Judy, to Sharon, to Rebecca, and to Mervi. And thank you, dear fellows. You fellow fellows have been making this such a special year, and it's been really wonderful. So, I want to tell you about one of the things that I've managed to do this year, which is to write a popular science book, as Judy mentioned. And this book is called The Society of Genes. I've written it with my very good friend, Martin Lurcher, and it's being published by Harvard University Press, and Thomas Lubien is here. And for me, the story starts when I was an undergraduate at BU. I was studying computer engineering, and I just came upon this book called The Selfish Gene. I was reading it, and it completely changed my life. I just couldn't believe that I had not heard of this earlier. I was totally enamored by it. I left computer engineering, I became a biologist, and I never looked back. And what this book is about is the language of inheritance. It says that there are these units of inheritance called genes. I'll tell you more about that in a minute. And that these genes are acting as though they're selfish, and that these genes are passing down the generations. And now, many years have passed since this book has been published. We know a great deal. And what Martin and I try to do in this new book is to talk about how the best way to view our inheritance, our genetic inheritance, is as a society of these units, as a society of genes. And it's kind of like Adam Smith wrote about the efficiency of the marketplace. He wrote, "It's not from the benevolence of the butcher, the brewer, or the baker that we expect our dinner, but from their regard to their own self-interest." So the efficiency of the marketplace follows from the self-interest of each individual. Well, the same thing happens in the society of genes. In our genes, we have these genes that are forming amazing relationships of cooperation and competition, and together they are managing to survive. And what this book is about, and that I'll tell you about today, is about their strategies. How do they do it? So one fundamental aspect of the society of genes is that it evolves. Evolution is at the core here. And evolution is a bit of a dirty word, in the US at least. I don't know of any presidential candidate running for 2016 or in the past, maybe you'll think of some, that have espoused evolution. Most of them say that they don't believe in evolution, and they're not alone. 40% of Americans do not agree with the sentence, "Human beings, as we know them, developed from earlier species of animals." 20% are on the fence, only 40% agree. In other countries, the situation is different, but clearly there's a lot of ways to go. And clearly, biologists such as myself are to blame. We need to present the evidence better. So in 5 minutes, I'm going to show you some evidence for evolution, okay? Tell me if you like this. So here's an experiment done by my friend Roy Kishoni when he was at the Harvard Medical School. Here's the experiment. He took a big plate, and he put really yummy food, at least yummy for bacteria, on this plate. Okay? And now here's how the plate is done. At the beginning, it's just the food. But then there's one layer where there's antibiotics. And then a layer just below it, 10 times the concentration of that antibiotic. So a lot more antibiotics. And then 100 times and 1,000 times finally here. And then he seeds bacteria. He just puts some bacteria at the very top. And here's what happens. So you can see the bacteria are growing, they're growing. But at a certain point, they reach the front. And this is the front where the antibiotics are. They want to go there. There's yummy, yummy food there, but they can't. They just don't have the ability to go further. They die as soon as they set foot into that territory with the antibiotics, at least for a while. If you keep playing the tape, though, what you see is that there's some bacteria that now start to grow at some point. Isn't this amazing? It's not everywhere, just at one point. And then later, there's some other parts that catch up. You see they're also growing. And now they're growing again. And they grow until they reach the next front. And then there's another pause. Oops. And if you play the tape still, when they get to the next front, again there's a pause, until some bacteria, again, manage to break through to the other side, to the place where there's 10 times more antibiotics. And you see, at some places, it stops. These guys, they want to go there, but they just don't have the ability, at least for a while. And then they keep going, until at the very end, you have these bacteria that have now occupied the entire plate, even the parts where at the beginning they couldn't do it. So what has happened? How has these bacteria been able to do this? And I can tell you that if you now take this bacteria, sample from here, and put on a fresh plate that's situated in the same way, it doesn't have to pause anymore. It will just migrate right through. They're resistant. They now have achieved this change. So what happened? In order to tell you what happened, I have to tell you a kind of secret ingredient that these cells have in them. And that secret ingredient is DNA. So DNA has been introduced. I'm sure everybody's heard about it, but I'd like to just introduce it again because I think it's so lovely. And let me tell you how I introduced it to my son's third grade class. You know that was coming? So I came to the class and I gave each student a strawberry. They put each one, the strawberry, they put it into a plastic bag. They mushed it all up, and then they filter it. So it's now like just the clean, juicy mush over here. Then you add alcohol, and then you could spool off, just like when you're in an amusement park and there's a cotton candy, you can spool off this white fluffy stuff, and that's DNA. So this DNA is not an abstract concept. It's a really physical thing. You can hold your hand onto it. And here it is. And if you had a microscope that's good enough, you could see that it's a one long string. This physical thing that we got out of the strawberry, it's a big string. And what the genes are units along this string. That's what a gene is. It's not a mysterious, abstract thing. It's a physical address on this string. So here's gene A. It starts here. It ends there. Then comes gene B. Starts, end. Then comes gene C, like beads on a string. And when you look even deeper, when you zoom in further, you see that it's a molecule that has, you would expect it's such a long molecule, so complicated. How do you know what it is? Well, really, it's like a language where instead of 26 letters, you have just 4 kinds of molecules, and they form a little permutation, a different order. So we have one kind of molecule that we abbreviate as T, and we see it throughout. We see a T here, we see a T there, and really, you realize that this is an informational molecule. This is a molecule that you can now you can abstract the information from it and summarize it as a string. So you can imagine my fascination of this when I was a computer, and used to be a computer engineer, and now I can get the sequence from this DNA, and that sequence holds the information. And now, if I had the complete set of sequence from the strawberries, I would say that I have the strawberry genome. That's what the genome is. It's just the DNA molecule. Okay, so now when we know what this DNA is all about, we can go back to the experiment and understand what happened. The crucial thing to understand is that the sequence can change. The DNA molecule is a molecule. It's a physical thing, so it can break, as we'll hear about next week from Leona Sampson, and the changes can occur. So imagine that you're writing up a sentence, my kingdom for a horse, and you just typed it wrong and you have a typo. That typo is unfortunate here because it really changes the meaning. Same thing for DNA. It's written in this 4-letter code, and you can have instances either because of changes from the outside or mistakes from the inside, but changes can occur, and we call this a mutation. All right, so how does this explain the Kishoni experiment? Here's how it explains it. At the beginning, we put, let's say, 1 cell. Remember at the very top? It divided. When it divided, it actualized its dream, which is the dream of every cell, really, which is to make 2 cells, right? And then each cell then made more cells, and it became 1, 2, 4, 8, 16, 32, this exponential growth. You got millions of cells. And the thing to realize that those cells are not identical. They're not identical because nothing in life is perfect. And every time you replicate the DNA, inevitably there are going to be some mistakes. So imagine that you have this strip of all these cells, and they're all slightly different from one another. And now you've got power in numbers. Why do I say that? It's because let's say there's one thing that a bacteria can change. And if it does that, it can now survive where the antibiotics are. Well, that mutation is really rare. What are the chances that exactly at this part of the book of its life, it can change it's really rare. But remember, there's many, many different kinds of bacteria. There's many, many copies of them. So now it's no longer a rare thing. It's actually expected that one of them is going to have the right mutation. It's like there's a lottery and you buy a ticket. What are the chances that you're going to win, right? But what if you buy all the tickets of the lottery? It's the same thing here. This bacteria, they're buying all the tickets. So of course one of them is going to win. Okay, so now what happens? You see, when they get to the edge, many, probably nearly all of the bacteria cannot survive. They all die. That's why there's an X here. But one of them has what it takes to now survive in the antibiotics. And that one does a really important thing. Again, actualizes its dream to make 2 cells, but the 2 cells have passed on what was so important, right? Because you made a copy. So whatever that chance mutation was, that gets inherited. This inheritance is a crucial part. And it's like when you're making a copy of a piece of paper, that copy might now have a blemish, right? And if you take that copy and make another copy out of that, that blemish will be inherited, right? Because it's there. Same thing here. This cell that managed to go into the uncharted territory can now make copies, and those copies are also able to live live there. And again, how does it manage to evolve the ability to live in more concentrated antibiotics? Again, there's power in numbers, and the whole thing plays out again. So what we have here are the 3 conditions for a universal law. And this is the law of natural selection. What we've just gone through intuitively is exactly what Charles Darwin discovered. He discovered it not in bacteria, but in animals, when he surveyed around the world with the beagle. But it's the same principle, is that whenever you have a population of stuff, and that stuff has variation, the variation is heritable, and that trait that it's heritable in can have some implications for the fate of the cell, then those 3 criteria are met, you must have natural selection. You must get, in this case, bacteria that have adapted to the antibiotics. So what I think is really beautiful about this is, one, that Darwin discovered this in the late 19th century without knowing anything about genetics, right? Second, I think it's amazing that if we ever find life anywhere else in the cosmos, we can be pretty sure that it's going to be very, very different, right? I mean, the whole concept of us trying to communicate with aliens seems ridiculous to me, because what are the chances that they're going to be humanoids? It's completely far-fetched. It's not far-fetched that there would be life, though. That there would be something that can self-replicate, that seems okay to me, because of this general principle of natural selection. Everything would be different on life we discover somewhere else, but the principle of natural selection will be the same. That's why it's a general principle. It's applicable far beyond this bacteria. It's applicable to the evolution of animals, to cancer, as Judy mentioned, everything. I love this principle. Now, now here's the book that I talked about, The Selfish Gene. So what The Selfish Gene did was really bring evolution to the modern era. And what Richard Dawkins said so provocatively is that, hey, you know what you are? All you are is just a vehicle, a survival machine, he called it, for your genes. You are just the living embodiment of the strategy of your genes to make more copies of themselves. And you know what? That's actually the truth, I think. I mean, it's always difficult. It's always surprising. As Dawkins himself wrote, he could never really get used to this idea. But that is, at one level, what has brought us here. Where it will take us, that I'll talk about at the end. So really, what he said is that the gene is immortal. We come, we open our eyes, we live for a few decades, and then we must close our eyes. But the gene, think about the gene. It keeps going. It's skipping down the generation. And you can compare the sequence of one gene to that sequence that was present 10 million years ago, and it's pretty much the same piece of information. It's immortal, he said. And that idea really captured the imagination of myself. It also captured the imagination, let me skip this. It captured the imagination of my friend Martin Lurcher. And together, what we wanted to do is to kind of bring this idea now to the modern era. This book, The Selfish Gene, was published 40 years ago. In the time since, we have the knowledge of many, many genomes. You know, when Dawkins published The Selfish Gene, not a single genome was published. Not one, not even for a virus, nothing. He had no idea what a genome looks like. And now we do. Now we can learn about the strategies of the genes, and this is what we set out to do. So we wrote this book, and it's meant for a popular audience. It's for people that may not have heard of DNA before, have no biological training. And what I'm going to do now is give you some bits and pieces of that book. Okay, so let's get started. This is I think, the best way to start to think about the strategy of the genes. So remember I said that Darwin didn't know about genetics at all? That's really a shame that he didn't know, because on his desk, there was a paper by Gregory Mendel, and he just never read it. And he was not alone. Basically, the entire scientific community ignored this paper, and it had to be rediscovered at the dawn of the 20th century. And what Mendel did was really amazing. Mendel was an Austrian monk, and he wanted to work with mice, but the head monk wouldn't let him work with mice because it was disgusting. So he convinced him to work with peas, because peas are at least edible, and it would be useful, some use. So Mendel worked with peas. And let me summarize one thing that I think is most striking and really cast a long shadow over the history of biology, which is that he really tried to say that there is a gene for something. So what I mean by this is that he didn't call them genes. That was left for a future scientist to coin. But he called them heritable units. And he said that look, this is a unit of inheritance that controls the color of the seeds of the peas, yellow or green. And here is a gene for the shape of it. Is it wrinkled? Is it round? So he really found what he thought was a one-to-one linkage, where for every gene, there is one function. And I think this really still has an influence on biology today, and it's proved really valuable in many, many respects, where when we set out to study a gene, we want to know what is it for. When we study a trait, we want to know what is the gene for. I mean, what is genetics? We have a mutant, and we try to see what, an interesting mutant, we try to see what the gene responsible for that is. And the thing is that this one-to-one mapping does not turn out to be true. It's not true at all. Rather, the situation is more like it is in Hollywood. So have you heard of the game called 6 Degrees of Kevin Bacon? The way it works is this. You're at a party, someone shouts out the name of an actor, say Robert Downey Jr. And what you have to do is you have to connect that actor to Kevin Bacon. And so you would say, okay, Robert Downey Jr., he acted in the movie Shortcuts with Julianne Moore. And then Julianne Moore, she acted with Kevin Bacon himself in Crazy Stupid Love. So that's 2 degrees away that Robert Downey Jr. is from Kevin Bacon. He's got a Bacon score of 2. And of course, this works because Kevin Bacon is one of these actors that acted in a bunch of movies. And each movie, he acted with a lot of actors. So any actor is somehow connected to Kevin Bacon. And of course, Kevin Bacon is not that special. It's going to be the same if you choose any other actor at the center. It would still work. And genes act really, really similarly. Why? Because if you look at a particular function, there are many genes that work together in a concerted fashion to bring apart this function. There's pathways of genes, complexes of genes. They all work as super entities to bring about a given function. And if you look at what a gene does, it typically is participating in many, many functions. So really, you can play this with many different genes. Let's say PAL1. When I was in Craig Hunter's lab as a postdoc fellow, I studied this gene. And it functions in muscle. You can see that it's connected to a particular function to many other genes because they're all in a concerted way functioning in this particular tissue. And also, it functions in the development of skin and also in neurons. So this is one gene, but it has functions in many, many different biological processes. And this is the best way to understand, I think, the society of genes. It's all these interconnected relationships. If you now zoom out, you don't just look at one gene, you look at many genes, well, then you see that there's a really, what's called now the hairball in biology, because doesn't this look like a tangled mess? This tangled mess corresponds to how we see the relationships among genes as. Okay, so that's one thing to know about the society of genes. They're not, as you would expect from the Dawkins metaphor, just a bunch of selfish genes. They're actually all collaborating together, and they're forming a tight relationship. Okay, now I want to tell you about how genes come in and come out of the society of genes. Dawkins wrote the gene is immortal, right? But that's not really true. It's actually the society of genes that's immortal. It stays alive, but the genes come, the genes go. Genes are born and genes die. And I want to tell you a story about the birth of genes. I want to tell you about a story about the death of genes. Okay? Here's a story about the birth. So I think it's kind of like a human society. I come from Israel, and there at the start of the 20th century, there was a social construct called the kibbutz. And the kibbutz is a kind of idealistic communal living where all the kids live together, and there's one bank account because they're all working in one factory to produce one item. They're all working together, kind of like genes in a genome, right? They're all working together. And over time, this grows. More and more more and more people are incorporated in the kibbutz, right? There's more kids, so there's kind of like a geometrical growth of the kibbutz. And you might ask yourself, but if they have a factory, so there's a number of jobs in that factory, and now you have more people, how do you reassign the jobs? How do you do that? And one way you might do that is to do what biologists call subfunctionalization, which is you take one job and you divide it into sub-jobs. So let's say one person was doing customer relations. You could divide it into customer relations for the north of Israel and customer relations for the south of Israel. You divided it up. Same thing happens in the genome. This is a story of how a new gene comes about. So the story is about hemoglobin. If you didn't have hemoglobin, you wouldn't be able to listen to me right now. Hemoglobin is crucial. The product of the hemoglobin gene courses through your veins, grabs the oxygen from the lungs, and distributes it to each and every one of your cells. And the thing is, you don't just have one hemoglobin. You have multiple hemoglobin genes in your society of genes. How do you have multiple? Well, there are sometimes accidents, special mutations that generate another copy, a new gene. And this is kind of like a society where you have new people, and how do you now give a new function, a new job to this new gene? Well, what happens a lot is that there's subfunctionalization. Here, these two genes, there's adult hemoglobin, I'll call it, and fetus hemoglobin. And it turns out that the hemoglobin that you are using right now, as you are listening to me, is one particular hemoglobin. But when you were in your mother's womb, you were using a different kind of hemoglobin. It's a different gene. It can grab the oxygen more avidly. And if you think about it, you need to have a different kind of oxygen when you're in your mother's womb because you were grabbing oxygen through the umbilical cord, right? And it had to already travel from your mother's lung to the umbilical cord, and then you had to grab it again. So it was a special kind of subfunctionalized, kind of like the customer service north-south, kind of different variant of the hemoglobin. And this is one way of incorporating new genes into society, right? This is the birth of a gene. Now let me tell you about the death of a gene. Some people think that humans are not evolving anymore. Some people think that our society is fixed, but that is not true, right? Because our society answers the 3 criteria for natural selection. There's variation. You could just look around the room, see the variation. We know it's heritable, right? And a lot of it corresponds to how successful we are in leaving more kids. So it's still happening. Evolution cannot stop with us. And let me give you a specific case of evolution in humans. If you are lactose intolerant, you've probably been made to feel as though you have some disease, but you really don't. You don't have a disease at all. What you have is what used to be the normal state. But then, only recently, when I say recently, I mean 10,000 years ago, there was the agricultural revolution. And humanity started to grab, as an extra source of energy, energy from cow's milk, right? And if you have cow's milk, that's great. I mean, we're all mammals. We all get milk from our mother when we're babies, but all of us turn the mechanism to break down milk after the weaning is done, so we don't use it anymore. But if you think about it, if you could just change it and have it so you can digest milk throughout your whole life, then you can have access to a rich nutrient. So that is exactly what happened 10,000 years ago. There was a new a new gene that was born. And this gene, it's actually just one single letter change. One single letter change difference from the gene that we have to break down lactose. And over time, it's gained in popularity. I mean, right now, 25% of humans have the lactose tolerance throughout life version of the gene. It's very popular probably in this room, because a lot of us here are from European descent. But if you were to go to Asia, it would be very rare, actually, to have that version of the gene. And this is the present. And it seems as though the other gene is on its way out. And what could be in the future? Maybe this gene is actually going to leave the society of genes. It's going to die. And this happens all the time. Genes come in and genes come out. So what I should say specifically now what I mean by the society of genes. The society of genes is just the sum total of all genes in a given species. So if all of us just stand around here in a circle and we all toss in our genes, if we all do this, then at the bottom there, that would be the society of genes, right? And here's maybe a more pleasing way to look at it. Imagine that you go to a city center or a town center, and there are 4 shops, 4 kinds of industries in that center. There's an eyeglasses shop, there's a bookstore, a shoe store, and a cafe. And you can go to a different city center now, and there'll be a different set of shops. But it's going to be, maybe a different glasses store, but there's still going to be a glass store. There's maybe a different kind of cafe, but there's going to be a cafe. The only question is it going to be a Peet's Coffee or a Starbucks, right? So you can imagine that in our society, there's competition in a given industry. And Starbucks is now, really rising in popularity, and maybe other cafes are going down. And the same thing is happening in the society of genes. For every one of our genes, and we just saw this with the gene that helps break down mother's milk, there's multiple copies of it. Like there are the people here that are lactose intolerant have one copy of the gene. The people that are lactose tolerant throughout adulthood have a different copy of the gene. And you can see that here. They have different copies of it. And these copies are actually competing with one another. So there's competition in the society of genes. All right. I want to actually move on now to another strategy of the gene. This I think we could not have predicted at all if we had just read the selfish gene. This is completely new to us. And this is that sometimes in the history of life, there can be huge changes, not just one new gene, not just one gene dying, but you can have an entire merger of 2 societies. And here's a story. I know it's going to sound controversial. I apologize in advance, but I would like to say that there are 2 kinds of cells on this planet: the boring kind and the interesting kind. I'm sorry, I apologize. The boring kind are the bacteria. They're small. They have interesting metabolism and all that, but come on. Look at these guys. Look at the eukaryotes. These cells are amazing constructs. They're huge. They allow for all the differentiation. The fact that you have skin, muscle, neurons, it's all derives from the ability of you having these large cells that can take on these different forms and allow this function. So the question is, why? How are these so awesome? And these, so here's, here's what happened. Here's what happened. These guys are really constrained. They cannot get bigger. Every time they try to get bigger, their volume rises in a much higher rate than their surface area, right? It's a different equation. This one rises faster. So whenever you have a bacteria that tries to get bigger, it won't be able to energetically sustain it. Why? Because the energy derives from a gradient that it sets up on its membrane. So its membrane, the membrane for a bacteria is really crucial. That's how it's going to get energy. And that's why it can't get bigger. It's stuck. So then you might say, well, how do these guys do it? How do they get energy? These guys have an amazing trick, a radical, pioneering trick. The trick is they don't use their membranes at all. What they do is they have these organelles called mitochondria. And the mitochondria, they make the energy. They're these powerhouses. And you know what? Where we got these mitochondria? Where did our cells get these mitochondria? Where 2 billion years ago, there was a really crazy event. One bacteria was swallowed up by a different kind of prokaryote, right? It was kind of like a Pac-Man thing. Like one thing was swallowed up another, and then they proceeded to live using with a symbiotic relationship. And that's what remains to this very day. Each of our cells is really a symbiotic relationship with the mitochondria, which used to be independent cells. And I should tell you that it might have been controversial what I said about the bacteria being boring, but it's not controversial at all what I'm saying about this model. This is accepted by, it remains unchallenged. And one really important piece of evidence, for example, is that the mitochondria still have their own genomes. So when we say our human genome, sometimes we're really slighting an important source, important contributor, because our mitochondria, that's an important piece of the puzzle. They have genomes too. We should count the mitochondrial genome as well. So I think this is an important piece of the puzzle for the Society of Genes, because it says that sometimes you can have these crazy mergers, two mergers of cultures, and you can think what kind of analogies you might propose for humanity, but sometimes mergers can have really dramatic consequences. As a final story about the Society of Genes, I'd like to tell you about freeloaders. So you might have noticed that in any kind of society that you have, any kind of set of relationships you have, there are always the freeloaders. And you say, why are there always freeloaders? Why does this happen? And one freeloader that comes to my mind is Kramer. Remember Kramer? He was the guy that's always borrowing stuff from Jerry. And one time, I remember he actually did something useful. Jerry needed a blood transfusion, and Kramer gave him one. But then Jerry was really freaked out by this, and he said he can't get the image out of his mind that Kramer's cells are there borrowing stuff from his cells. So you might say, are there also freeloaders in the genome? And it turns out there are. The Society of Genes is packed with freeloaders. That actually, to a first approximation, what our genome corresponds to is a really runaway phenomena of freeloading. And here's one kind of freeloader. There's a freeloader of the type line. And what these lines do, they're genes. You know, they have a place on the chromosome, like we talked about with the strawberries, and they encode a program that effectively says, hey, copy, paste me. Copy, paste me. And that's all it does. There's machinery here that it encodes, there's proteins that it has to go through, but basically all it does is, the only thing, it doesn't contribute at all to breaking down lactose, doesn't contribute at all to building up muscle. All it does is say, hey, copy me, copy me. And it works. It works. It works in the sense that 20% of your genome is this. Imagine every time imagine you get like a cut and new cells have to be formed, those cells that are being formed, a lot of time is being spent in just copying the freeloaders. And the freeloaders, as it turns out, also have freeloaders. There are things called sign elements, and the sign elements, what they do is they don't encode anything. They don't even go to the trouble to encode this copy-paste strategy. All they do is they make themselves look like the thing that needs to be pasted back. And these guys are super successful. There's over 1 million copies in your genome. 1 million copies of your genome. We don't even count them. They're genes, right? But when we say the human genome, we don't say you have 1 million genes, right? We don't even count them because we know they're just freeloading. So to summarize what I told you, I've shown you aspects of the Society of Genes. I've had to summarize a lot because it's a short talk. But we said that these genes are cooperating and they're forming a network of interactions. I showed you how genes come in and come out, die of the Society of Genes. I showed you how societies can merge in rare cases, and these have important implications. And I've showed you how the Society of Genes can be taken advantage of with all the freeloading. Now, to conclude, I want to tell you about something that happened when I was a postdoc here. It was great to be at Harvard, because when you're at Harvard, you don't have to go see Bill Clinton. Bill Clinton comes to see you. So there was I think it was 2008, Bill Clinton came and he gave a talk at Harvard Yard, and I was one of the many thousands of people that were sitting there. And he said something that really grabbed my attention. I found this so inspirational. He said, you know, when I was president, I funded the Human Genome Project, the project to sequence the DNA of humans. And these scientists, you know, they kept getting more and more money out of me. They kept saying, oh, you know, we spent too much, we need more money. I said, take it, just do it right. In the end, it was $2 billion. And it was worth it. Why was it worth it? Because I learned something amazing. I learned that between any two people, we have 99.9% identity in the genome. Think about all the wars, misunderstandings, all these things that get in the way of peace, all because of the 0.1% that we choose to highlight. And I thought that was really impressive. And I think he thought it was really impressive too, because he's been citing this in many, many speeches, hundreds of speeches. Hillary Clinton has also been using this. And I want to tell you something. I want to tell you that it's actually really dangerous and really kind of misinformed, this whole argument. And it's in a really surprising way. I call it, Martin and I call it the Clinton paradox in the book. And why is it called the Clinton paradox? There's another side to the 0.1%. Our human genome has many letters. To be exact, it has 6 billion letters, right? 6 billion letters long. It's a really long encyclopedia. Now, 0.1% of the 6 billion amounts to 6 million. So any two individuals, 6 million differences. 6 million differences. That doesn't sound, that doesn't sound so small anymore, right? And so what is it? Are we mostly the same? Or are there so many differences that you can pretty much understand what's going on, why people cannot generally get along? And in the chapter, we enumerate these 6 million differences. We go through them. No. Not one by one. That's a lot. And we come to the conclusion, actually, that this is silly. Well, first of all, you know, what if Clinton had gotten different information? What if we had told Bill Clinton, you know, humans and chimpanzees were 99% identical? That also sounds pretty good, 99% identical. And really, it's dangerous to play this numbers game. And I'll show you one really extreme example. And the example is this. Let's say all of humanity had exactly the same DNA, except for one difference. Half of the people had one variant, and the other had the other variant. So there's one difference that separates all of humanity. And let's call this what has been called a green beard effect. We'll say that this gene that separates humanity develops a green beard in all the people that have this gene. And it has two other attributes. It can detect if someone else has a green beard. And if it does, then we'll treat them differently. We'll help them out. In other words, there'll be a brotherhood of green beards. And you can see how this plays out. Even with this one difference, it's already enough to separate an entire species. So really, it's pretty silly to break down the argument of to make a cause for human peace based upon these numbers. Really, what our position is the following: to act in such a way that highlights your difference with another person. If you're doing that, really, you need to consider that you are acting for a minor part of your genome. Your genome is dictating this behavior, a tiny part of your genome, and you're acting in such a way. And what you have to remember is that you are not in the heart and mind of your genes. The genes don't really care about us. The genes are just building us, and they want to keep going. So we are not in their interest. So why should we act in their interest? Why should we highlight the difference based upon just the wishes of a tiny fraction of the genome? Really, and just to conclude, I want to say that in our genome, we see all the history. We can track how we got here. And I think it's really amazing that we are part of this species that, any other species also has a society of genes. But we're unique. We humans are unique that we have the society of genes, and we realize what they want. We realize that they want us to fight, in a sense. And we can decide how we are going to deal with it. And I think that's a pretty unique position to be in. So thank you very much for listening. Thank you. Yeah, Renee.
Thank you for that really stimulating talk. I wonder if we could go back to your first slide where you're showing the bacteria.
Oh, actually,
you don't have to if you don't want to. I'm just curious if you could talk about.
It's a max. I don't know how to.
If you could talk about maybe biologically where or why evolution does not happen in certain cases. So, I mean, you sort of are explaining how it does happen, but I'm really curious, why doesn't it happen in cases where things are, for instance, in the body that we no longer need, or in case of sort of, I guess, trying to fight cancer? Why is it not happening?
Oh, so so the question is, why is evolution not happening in some cases where we expect it to happen, like parts of the body hanging around, or in cancer?
What does it mean for it not to, for a thing not to evolve?
Okay, I think I understand. So, some cases, you just don't meet the criteria for natural selection. If you have a population where there are differences, and the differences are heritable, but they're just of no consequence, you will not get natural selection. So one example is that, let's say I had a blue hand. And normally you would think that this might harm my chances of finding a mate, because I have this blue hand and I'm really self-conscious about it. So, but what if I manage to cover it up all the time, so no one knows about it? So there is variation. It is going to be hereditary, but if it doesn't change my odds of having kids, it's actually invisible to selection. So that's one case where. But I think you were hinting at something else where there's parts of the bodies that sometimes people talk about the appendix. Why do we still have the organs that we may or may not have a need for? Well, so one thing to remember is that evolution takes time. I on purpose showed you the example of bacteria because it's happened so fast. We just in a day make a movie and I can show it to you and everybody's happy. And that's because bacteria divide every 20 minutes. But for humans, when I talked about the lactose, that's something that happened 10,000 years ago. And we see it in a gene that's doing very well, but still doing very well only means it got to 25% of the population. There's still a long way to go. So sometimes it just takes a long time. And then you mentioned cancer.
Well, yeah, because some cells do not adapt, right? Some cells don't figure out how to get to that next level. So what is that about? Is it just you're not smart enough, or?
So with cancer, actually, we have a beautiful case, as Judy alluded to in the beginning, a beautiful case of evolution. It's not beautiful to the person, to the patient, obviously, but it's beautiful in that it really is an evolutionary process. And the cells that are evolving are the cells that are evolving are getting a fitness. Are getting an advantage over the other cells because they can now make more copies of themselves. So really you have this population of cells that are becoming different, and they're passing along this difference, and they're outpopulating our own cells. And there's lots of checks and balances that our good society of genes have put in place to stop that, but sometimes it doesn't manage to do that. Yeah, Jennifer?
Well, thank you for such a wonderful talk. And it sort of pained me to listen to it, but it hurt so good. And the way that it was, I thought, if for just once, when I was younger, I had a teacher in the sciences, I'm serious, maybe I would understand just a little bit more of our natural
that's why
world.
Okay.
So I want to thank you. But because I didn't have a teacher like that, I became a humanist. And as a humanist, I encountered Thomas Kuhn, Evelyn Fox Keller, and those are just two names who helped me, at least, and I'm helping my students, and perhaps conducting some malpractice in doing so. So teaching my students their ideas, which were that science, the language that we use to talk about science is not neutral. You know, we don't use that language by nature or by necessity. We're human beings, and they're social. Our language is social. So our language for accessing our world and for describing our world is social, is bound up with time and place. And so I'm wondering then about the language that you and Martin, right, are choosing, and that process to call something, let's say, a freeloader. You know, in a moment, right, when that's kind of a charged word. So this is not a provocation, it's just a question, which is, did you think about different words that were on option to describe some of the processes?
Right.
And did you have other words that you played with that didn't quite do it?
Right. So did you see, actually, when I answered Renee, for a second, I said fitness, and I froze because I don't want to use that term fitness. I think it's not the... Martin and I did think a lot about the terminology, because I think, actually, there's the other side of it, which is jargon. And a lot of scientists use jargon, you know, all too willingly, and it really just obscures our work to just a particular sect. So we really wanted to make it more popular. And we did sit and try to think how to write about it. And I think some people would object to certain ways that we wrote about it. For example, universally, we anthropomorphize the genes. We say a gene wants to do that, right? And of course, we don't really think a gene wants to do anything. Really, that's just shorthand for a gene is acting in such a way, you know, it's programming a certain mechanism that makes it look as though it wants something. But that's too cumbersome to say, so we say it wants to do it. And I sometimes think that it's this costs and benefits, and if some people are able to get the concepts better because we make analogies to societies, I think overall there's a net gain. And you mentioned the humanism. I want to say that I read E.O. Wilson's new book, and he says something that I really liked, and I really think of now when I think of the humanists, which I've been in contact with now and now have a face, which is that if E.O. Wilson writes in his book, if aliens, you know, these aliens are completely different but still have natural selection, land on Earth, and they want to talk to people, they won't ask us, they won't really care about our science. They won't really. They don't really care. I mean, because presumably, if they can come to Earth, they already know about relativity. They obviously know about natural. They don't care. But the humanisms, that would be really fascinating. All those science, like the language and all that would be really fascinating to them. So I have a new respect for. Yeah.
I have another question about language, which. It's really striking that Dawkins talked about a selfish gene. And I'd have to think more about the timing in the mid-70s and all of that is interesting. But you kind of talked about both the selfish gene and also a cooperative gene. So does this mark a real departure from the kind of, let's say, Dawkins-Smithian framework to something else?
Okay. Does talking about cooperation among genes mark a certain departure from Dawkinsian framework? Not at all. So Dawkins, he was well aware that cooperation is just one really good strategy to be selfish. He was well aware of that. And so what we're saying is not new. I mean, we document it much better. We show it in exquisite detail with all its beautiful angles. But it's not new. He would readily agree, and he would say, "But what do you mean? I said it myself." So that's not new. I think what is new is that we're really seeing the strategy among the genes. I think something like the mergers of entire societies, that was entirely unanticipated in Dawkins's time. Yeah.
So I'd like to come back to that slide where you showed the little boring bacteria.
The boring ones?
And no, and the big, interesting eukaryotic cell with its nucleus. And
I have it coming,
membranes.
yes.
You got me sitting.
So,
Oh, yeah.
my point is, I have a question to what you think of the following, and that is some people have put forward the idea that multicellular organisms like us, with all those lovely cells with nuclei, we have 10 times more, or is it 100 times more bacteria
Right.
living within us. And one theory is that really bacteria drove the evolution of multicellular organisms so that we can be incubated to grow those bacteria so that they can survive.
Yeah.
So if you turn it on its head, what do you think about it?
Yeah. Okay. So Leona is saying, let me see if I have you right, that these bacteria shouldn't be ignored because even in our cells they overpopulate what we think of as our own cells.
Exist because of the bacteria.
We may only exist. So I would say this. I would say, look at what I've said. I've said our cells are so amazing because they have an internal bacteria, right? So it really is all different kinds of bacteria, right? There's all life forms. Actually, I didn't say it, but on a global scale, what's going on here on this planet in terms of life is the viruses versus the cells. That's the big story, actually. But if you now look at just the cells, there's just different kinds of bacteria. And we're the special kind of bacteria where it's two bacteria living in symbiosis. So I think that the importance of bacteria is, runs through what I'm saying too. And I just said that they're boring to wake everybody up.
I want you to go back to the cancer. The proliferation of cells isn't really the problem, it's the metastasis. How do you relate that to your society of genes? Because just expanding, making a tumor removed and so on, but the negative transformations, that seems to be the tricky part.
Right. Okay, so the question is that in cancer, there's multiple steps, and the final step is to break away from the tumor and conquer other places. These are known as metastases. So how does this gel with the notion of the selfish gene? Well, the way I think about it is, remember when we had the bacteria on the plate and we had different concentrations of antibiotics? With cancer, it's the same thing. I didn't get a chance to talk about it at all, but there's different steps. There's one step, which is, hey, stop listening to your neighbors. That's the first step, because the neighboring cells are always telling a cell to stop dividing. So the first thing is just, hey, don't listen to what they're telling you. Do your own thing. That's the first step. And there's another step that says, okay, now you're going to have to become stable as you're making more copies of yourself. And there's many more steps. And then the final step is now that you're going to have to acquire the ability to conquer new parts of the body. And why are these steps? Because if you think about it, that whatever group of cells can do that, they achieve an advantage. So that's why they do it. And the same reason why the bacteria keep going down. Why do they keep going to the other place? Because there's food there. They want the food. Whoever gets the food gets the green light to make more copies of itself. Same thing. If a tumor can now metastasize and go somewhere else, they have the green light to make more copies of themselves. It's just like the bacteria. So I say it's the same notion. It's just a set of mutations. Those mutations cause variation. They're heritable. They provide a fitness to those cells that can do it. And metastases is just one more step along the path. Oh, sorry, I haven't answered you guys. Oi, and then you. The first one.
Thank you. So going to the merger analogy. So on one hand, we can continue to cooperate more and more if we acquire more companies, more and more. So we get more cooperativity by increasing our genome size. At the same time, we're going to have to pay a price to replicate and to make more of that. So can we sort of think about, is it important to see what's going to win in the end? Is there a balance, or is it just we can continue on growing and at the same time keep making copies and so on, or at some point some will win?
What an interesting question. So the question is, can I've shown evidence that there could be mergers, but can you have more and more mergers, and what can limit the mergers? I think there must be limits on the mergers, because otherwise, the merger that I talked about, it happened 2 billion years ago. I had to go back that far to find an example. So it can't be that popular, but once it does happen, it seems to have a lot of consequences. I can tell you also that there have been other sorts of mergers. There have been like internal duplications. If you look at our genome, there's really good evidence that over time there were 2 instances where the entire genome got duplicated. So now you have twice of everything. And that happened twice along the last 500 million years. So that's rare too. Plants, they can duplicate a lot. So there it's more common for some reason. But there must be some limits. There must be some. I guess at a certain point, if you just have too much, it just disturbs the basic functioning of achieving this dream of getting 1 cell to 2 cells. But okay, you back there.
Clearly what we have here is an emergence going on. I mean, just look at ourselves coming from the slime and all of this other stuff. So there's got to be some properties that do this, and I've got to believe that at some level there's a math going on, just as the properties between the integers, the prime numbers. It's kind of like a hardwired system of math. I mean, 55 is the sum of the first 10 numbers, but it's also the sum of the first 5 squares. So there are these hardwired relationships you get. And my question is, because of that hardwiring and then its much later manifestation of great complexity, is there, do you feel, a teleology built in? Not that humans would have evolved on other planets, but that the very fact that things took the way they did, and that everything's exponential. Information, knowledge, computers, human population, we've entered in the last few hundred years, I don't believe, everything's going exponential. Do you feel that there's some teleological built-in at some core seed level that's causing this manifestation?
What an interesting question. I think you're really asking, are there kind of these hardwired rules that biological systems must follow?
Well, like a fractal, you have a very compact equation, yet it manifests in some very large thing. So, I mean, a human is just far beyond anything around, and what we create is even more beyond that. And the question is there a teleology built in at a seed level?
So
when it manifests, it just creates this unbelievable ordered complexity.
Interesting. I think what I would say to that is from time to time, physicists find biology a little bit disappointing because it's less like physics. We have less. Well, correct me if I'm wrong, but really biology is a kind of a historical science, in that there's constraints that occur historically, and then the future generations are constrained by them. So, for example, we have this DNA. We encode our information by DNA. There's no way that we can imagine. There's no way that it has happened, actually, that there is now a second kind of informational molecule. Like we have to live with DNA now. That can't change. And that's all because of something that happened 4.5 billion years ago. So it's really historical in that sense that there's an accident, it gets frozen, and we're constrained by it. Now, beyond that, there are laws. And I told you about the only real law that I know of in biology, which is that of natural selection. And it's got these basic assumptions. And if you have, sorry, I shouldn't say assumptions, criteria. And when you can meet those criteria, you must get natural selection. That, in my mind, is the only real principle, and everything else kind of derives from that. The mergers and the new genes and the old genes dying that I've told you about. That these are all kinds of like historical observations that we find. We're looking at how life has unfolded, but it all operates through this kind of I would say Darwinian mechanics of natural selection. It all has to play along with these rules. So that's the only real principles I know. Kamala?
So what do you think about editing the human genome?
What do I think about editing the human genome? So, I read a New York Times op-ed a few weeks ago about editing, and the argument there was that we shouldn't edit our own genome, because then we would be we would be changing our species. We would be changing our species. And that struck me as really odd, because that makes it sound as though there is the human species and we would be changing it. But really, but really, the human species is always changing. That's what we are. There's a society of genes, and things are always coming in and out, and things are always changing. So it's really a ridiculous idea to suggest that we would be, for the first time, changing anything. And I gave you just the example of the lactose intolerance as one change, but there's many more examples. So I think my position on editing, and I would have to think about it more, but it would be that if we see that editing the genome can serve some of our values, and I think one value that I could think of is that we want to minimize suffering. If we can somehow get rid of a disease and then there's less human suffering, I think that would be a good thing. So really, we would need to use common sense against not using it to only have girls or only have boys. That would be short-lived. Short-lived. Thank you, Pupa. Yes. Pupa, yeah.
So many things to say. So first of all, physicists are not always against biology.
Oh, yes.
So I embrace it wholeheartedly. And if I can use a teleological argument, one of the beauties of biology is that you actually can ask the why questions, which in physics you can never ask. There is no why. You can only humbly describe how the laws of nature behave. Whereas in biology, you can ask the why question, and why doesn't mean which God put it there to answer the teleological question, but it means what is the evolutionary damage of that particular behavior or mutation or whatever. The other thing I actually wanted your opinion about is about cancer. Have you heard the recent theory according to which, and this only came up in the last couple of years, so you may not have heard about it, but the story is that cancer cells basically go backwards along evolutionary time. And they revert to a time in which there was single cellularity rather than multicellular organisms, as we all are. Not considering the bio that you pointed out. So the proof that they provide for that is that, in fact, cancer cells are often resistant to hypoxia, which is why they end up being resistant to treatment often, because they're tougher cells, basically. They go back to a point in time where atmospheric oxygen was much lower. And in fact, because of that, they're harder to get. So how do you reconcile that theory? Have you heard of that? Do you like it? Do you not like it? Does it contradict anything you observed?
Yeah, good question. So what Pupa is saying that there is recent evidence that in the development of cancer in an individual, really what those cells are doing is they're leaving their posts at being like skin cells or liver cells, and they're returning to a more primitive state, at least in the timeline of the individual, to when they were an embryo. And so I have heard of that, and I'm actually studying something tangential to that, because as Judy pointed out, I look at many different kinds of species, and what I've noticed is this, that if you look at the genes that we know of as those that cause cancer when mutated, and you map them to when they function throughout the life of the organism, it seems that there's two kinds of sets. There's the set of genes that come on right as the embryo is starting to switch to becoming differentiated, and there's a set of genes that are on right when they're at the very beginning, when cells are proliferating. And it turns out that the proto-oncogenes, those are at the switch, and the tumor suppressors, those are at the beginning. So it really is as though, what is the embryo? The embryo is a fertilized egg that makes lots of cells and then puts them in their place. And what cancer is to really kind of go backwards. So we are seeing evidence of that, and it's an interesting point. Thank you. We have time for more?
Yeah.
Okay. Henry.
I have a broader question, which is, what exactly does it mean when you write that you've written this book with someone. I mean, in a practical level. I mean, this person is so far away. How actually what does it mean?
Right. So Martin and I, Martin is German, I'm Israeli, so it's like world peace in its own way. And we were really good friends. And how did we write this book? Well, first we taught it as a course. So all the time we were on Skype on the one hand and Google Docs on the other. So we were modifying, and we gave it as a course. So we were teaching. He was teaching it in Düsseldorf, and my students were in Israel. And then that's how we got the material. And then to actually sit down and write the book, we set aside 1 week every 2 months, and we would travel to some really boring place. Although people would argue with me and stuff, so I won't even say where it was. And, there would be nothing to do there but just sit down and write. And then the first time we did this, it was a disaster. Even though we knew exactly what we wanted to write, I would write a sentence and he would say, no, 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 and so forth. And then after 2 days, we just had a page and a half. So so then what we did was we would sit next to each other and we would just say, okay, you know what? You write the first part of the chapter, I write the second half, go. And basically, we would sit next to each other just to make sure that the other is not checking email. But that seemed to work out really, really well. Judy.
I want to go back to Presenon's question, which was, about if you go past self, you talked about the selfish gene, and then you have a cooperative genes. And when you answered Presenon, you said, well, cooperation can be a strategy. I'm wondering if you ever see any examples of altruism among genes. Do you know? Yeah.
Altruism among genes is sadly laughable. So that was really the seed. Sorry, that was really the basic insight of Dawkins. Dawkins would cite Hamilton's, who was a great biologist. Haldane, who said, another great biologist, who said, I would jump into the river to save 9 cousins. And the reason is because only, if a cousin is an eighth like you, then only if there's 9 of them that are drowning is it worthwhile for you to get involved, because then the net gain for your genes would be the same. So really it's all these mathematics of the genes. And if there was an altruistic gene that ever arose, that this gene would say, hey, always help this other gene, always help this other gene, then sadly, both computer simulations and experiments show that it wouldn't go far. It dies right away.
Well, we're out of time, so
Okay.
please join me.